Good morning to all students. Today we will discuss about the genetic code, which is an important concept to be discussed. And as we know that according to the central dogma, the genetic information flows from DNA to mRNA and then to protein. So this is the flow of genetic information. And it occurs first by from DNA to mRNA, which is takes place by transcription, and then through translation from mRNA to protein. So proteins are made up of amino acids. So how these amino acids are actually produced, or there are 20 different natural amino acids are there in human body and also in other organisms. So how these amino acids are arranged, what should be the sequence of the arrangement of these amino acids, and who actually guides this orientation. This is very much important to understand. And today this concept of genetic code actually unravel these facts. So how the proteins are generally synthesized, and how the amino acids in the proteins are coded, and how their sequence are directed, we will discuss today in our lecture. So first of all, we should know what is genetic code. So genetic code is a dictionary. So it corresponds with the sequence of nucleotides and sequence of amino acids. So if you, what will do with a dictionary? For a new term, we go in the dictionary, we search the term, and we get the corresponding meaning of this individual term. So likewise, the sequence is present in the nucleotides, in the form of nucleotides. So they are found in DNA or RNA. So this is our word, which meaning we don't know. This is the genetic code. Now means a codon. Now the sequence of amino acid which it codes is the meaning. So that's why it is termed as a dictionary. So genetic code actually it's a set of rules by which informations encoded in the genetic material, DNA or RNA. So DNA and RNA are, and as well as the proteins, they actually carries the informations. And basically the informations which are stored in the DNA and RNA, they are translated to the protein. So protein will get the informations from that DNA and RNA. And this genetic code is termed by a famous scientist, George Gamow. And here you can see is George Gamow, Watson and Crick, the Nobel laureate for the discovery of DNA structure. And this is the George Gamow, who postulated the genetic code for 20 standard amino acids which are found in our body. So now genetic code, as we have mentioned, genetic code is formed by nitrogenous bases. So there are 4 different types of nitrogenous bases: adenine, guanine, cytosine and thymine. So adenine, guanine, cytosine and thymine. Here you can see the bases are sorry adenine, A for adenine, T for thymine, C for cytosine and G for guanine. And in RNA we will get the four, this is the DNA basis, and in RNA we get A stands for adenine. In place of thymine in RNA we will get uracil, U, then cytosine and then a guanine. So these nitrogenous bases actually they orients in a fashion to codes for the amino acids. Now 20 amino acids are there. So from common sense we can say that for 20 amino acids there should be 20 codons. Now how many nucleotides should be there in the genetic code? So if we consider only A, it codes for one amino acid. Suppose amino acid 1. Now T, thymine, it codes for another amino acid A, amino acid 2. Then guanine, it codes for another amino acid and cytosine, which codes for another amino acid. So if this is the case, then only 4 amino acids will be there. But we know that there are 20 different amino acids. So a single nucleotide will not serve our purpose. Now go for 2 nucleotides. Suppose you consider A, then T together. If you consider AT, then AT will codes for one amino acid, then TA will codes for another amino acid. So for each combination we will get one amino acid. So for 2 bases there will be 2 combinations, and for 4 bases there will be 16 combinations. So for 2 nucleotides 16 combinations are there, and for 16 combinations, but there are 20 different amino acids. So 16 combinations will not fulfill the demand. Then we can consider for 3 nucleotides. If it is the case, then for 3 nucleotides there will be 64 combinations. And now the 64 codons can easily code for the 20 different amino acids. Now here you see this is the genetic code. This is the DNA which got the informations it got the genetic informations. This is the DNA. This is it got the genetic information. That is nucleotide sequence are there. Here you can see the nucleotide sequence. Now you can see that this ATG and CTA, then GGC, these are the different codons which codes for the amino acids. So they are made up of three bases. So genetic code is also known as a triplet codon. Now in our next slide we'll see the table, actually the table for the genetic code which represents 20 different amino acids. From this table you can see that in rows and in columns, and column that you can see that you see A and G are the codons, and in row also you see A and G. So now for this square you can get the 4 codons, 4 codes will be there, and for there are in total 8 squares are there, and in 8 squares if there are 4 codons then we will get the 64 codons. So this is a 64 codon table, and it is very simple to write the codons. Here you can see in the row and column it contains both row and column, and it contains the U. So you first write the first 2 codons, first 2 bases of the codon as UU. Then from the first codon up to the 4th codon you write this sequence U, C, A and G. So this will form your first box. Then for second box CU, then you write first the 2 bases as CU, then this sequence UCAG. Likewise you can get all the codons or all the codes for the amino acids. So here you can see that there are certain amino acids which are coded by a single code. As for example, the methionine is coded by AUG, and another codon is there which is the tryptophan. Tryptophan is also coded by this single codon which is known as UCG. Now there are stop codons. There are 3 stop codons. The sequence are UAA, UAG and UGA, and based on this codon the protein synthesis will get stopped. And here you can see that there are certain amino acids which contains more than 1 code, and leucine is an amino acid which got 6 codes. Here you can see this is the 2 code and these are the 4 codes. So in total for this individual amino acid leucine, we can see that there are 6 codes. So likewise these 64 codes are distributed among the amino acids, and 61 codes are distributed among the amino acids, and the 3 codes, the codons are the stop codons which actually terminate the protein synthesis. Now we'll see in our next slide the important concept, the codon and its type. So genetic code, as we have mentioned, it is nothing but the dictionary, and each codon consists of 3 bases which codes for the amino acids. There are 64 codons in total. 61 codons codes for the amino acids, and 20 different amino acids, and 3 codons are the termination codon or the stop codon, based on which the synthesis of protein will start. Now types of codon. So we can classify the codon mainly in 2 types. One is sense codon and that is signal codon. So what are sense codons? Sense codons are those codons which codes for the amino acids. And signal codons are of 2 types. One is start and another is stop. So start codons, generally methionine is the starting amino acids, and it is coded by AUG. So AUG is generally termed as the start codon. And signal codons, the stop codons are also covered under signal codons. And signal codons, actually there are also 2 types. We can also categorize or separate the signal codons or classify the signal codons into start and stop codons. So start codon is AUG and stop codons are the 3 stop codons, UAA, UGA and UAG. So they are also known as the termination codon. Now the terminating codons, they actually UAA, UAG and UGA, they are also referred as amber, ochre and opal codons. And initiating codon is AUG, it codes for the amino acid methionine. And in case of prokaryotes, there is difference. In case of the prokaryotes, the initiating codon is formilated, that is formyl methionine, and in prokaryotes, and in eukaryotes it is only methionine. There is no formyl group. Now what is anticodon? This is another important concept. There is tRNA, transfer RNA, which got these which got the anticodon loop and the anticodon. It is just reverse of the codon. That's just reverse of the codon. So tRNA is important because it brings the amino acids during protein synthesis. So that's why they are so much important. So they actually interact with the codon present in the mRNA. So there will be codon anticodon interaction in the ribosome during protein synthesis. Now, what is the difference between codon and anticodon? So codons are present in both DNA and RNA, but anticodons are always present in RNA, never in DNA. So codons are written in 5' to 3' direction, and the anticodons are written in just reverse order, that is 3' to 5' direction. Now anticodons of some tRNA molecules have to pair with more than one codon. So anticodon, they have to pair with more than one codon, because there are only 20 different amino acids, and the tRNA numbers are also limited. Now codons are sequentially arranged in nucleic acid strands, while anticodons are discretely present in the tRNA. Codons defines which anticodon comes next with an amino acid to create the protein strand. So it is decided by the codon which anticodon will come over. Now anticodon helps in bringing a particular amino acid particular amino acid during translation process. Now we come over to the characteristics of the genetic code. And genetic codes are triplet code. They are triplet code and commaless non-overlapping code. The coding dictionary, it is a coding dictionary, and there is degeneracy of code, universality of code, non-ambiguous code, that is non-ambiguous nature of the code, chain initiation code and chain termination codon. So these are the characteristics, and we will discuss some of the important characteristics of the genetic code here. So first of all, we should consider the triplet nature of the genetic code. So from the very beginning, we are mentioning that there are three different nitrogenous bases which actually makes the codon or the genetic code. So genetic code is made up of three nitrogenous bases. Now universality. So there are certain exceptions with this universal nature of the genetic code, but even though in case of prokaryotes as well as in eukaryotes, we will find that AUG is the initiating codon. AUG is the initiating codon, but there is also some exceptions that AUG sometimes codes for isoleucine in cytoplasm. So next characteristics is the non-ambiguous nature. So this is also very important. That is, one codon cannot specify more than one amino acid. That means for a single amino acid there may be more codons, but each codon can represent only a single type of amino acids. This is known as the non-ambiguous nature of the genetic code. Now next we come to the non-overlapping nature. So genetic codes never overlaps, as you can see here. I can draw here four. Suppose this is adenine, suppose this is adenine, this is guanine, this is thymine, or rather you can see here adenine, guanine, uracil, then cytosine, adenine, uracil, then uracil, cytosine, and guanine. Now here you can see that this is one codon, this is another codon, this is another codon, and this is not a codon because it contains a single base. So they are read as a set of three bases. So never you cannot consider a codon to be overlapping, because if it is overlapping, then you will find that the first codon is AGU. If it is non-overlapping, then the first codon is AGU, then second codon is CAU, then UCC. But if it is overlapping, then first codon is AGU, then second codon would be GUC. Here you can see that. Now first codon would be in case of non-overlapping nature, the first codon would be AGU, then CAU, then UCC. Now this is the non-overlapping frame. So if the genetic code is overlapping in nature, now you will find that the next first codon is A, G, U, then next codon would be G, U, C, then next codon would be U, C, A. So this never happens. Never happens. So that's why genetic code is non-overlapping in nature. Next, continuous translation would be there, and there will be, that means within the genetic code you will never get any punctuations. There will be no semicolon, full stop and comma, etc. So it would transcribed or translated continuously. Next, the code has polarity. As I have already mentioned that the genetic code is also read from 5' into 3' in direction. So the codon got specific polarity, otherwise different amino acids will be coded. So for example, in case of U, as you can see that if it is coded from 5' into 3' in direction, it codes for leucine, but if it is from 3' into 5' in direction, then it codes for the valine, another amino acid. So the meaning will be changed in the protein. So that's why there is the genetic code has a polarity. That is, it should, and it is always read from 5' into 3' in direction. This is another important concept, degeneracy of genetic code, and amino acid can be coded for by more than one codon, which is known as the degeneracy of genetic code. As I have already discussed, there are 6 different codons for the amino acid leucine, so there must exist degeneracy, and this degeneracy is due to Obel hypothesis. We will discuss later on. Now, what is Obel hypothesis? Obel hypothesis, this is another important concept. Now before that, I want to explain you the Obel hypothesis. It is a very important concept. Here you can see that the Obling actually occurs in the third base. That is, there is a certain degree of relaxation. That is, stringency is very less during the base pairing rule at the third base. So this is postulated by Crick to account the degeneracy of genetic code. According to his hypothesis, the first two bases, or the first two base of a codon pairs according to the normal base pairing rules. That is, G always pairs with C and A always pairs with U. These are the complementary bases. And with the last two bases and base pairing of the last two bases of the anticodon, these are the last two bases of anticodons, because anticodons are read from 3' into 5' direction. In this way, anticodons are read from read in this way, in that particular direction, and codons are read in that particular direction. That is, from 5' into 3' and an anticodon from 3' into 5'. The first base of anticodon would be G, and the first base of the codon is C. So here you can see that the base pairing of this first two bases and the last two bases, first two bases of codon and the last two bases of anticodons is as usual, normal base pairing. But base pairing at the third position, but third codon is Obel. It is Obel. That is, G normally pairs with C, but here the obal pairing occurs. G pairs with U. That's why, due to this type of base pairing, the obling occurs. Now we will see in detail. How obal hypothesis explains the degeneracy. Obal hypothesis explains the degeneracy of the genetic code. There is existence of multiple codons for a single amino acid. As we have mentioned earlier, although there are 61 codons for amino acids, the number of tRNA is far less than, that is around 40, which is due to obling. So how it is possible? There is not enough tRNA for the different anticodons, for different the codons. So if there is no obling, then you will get only 40 tRNAs available, as a result of which all codons cannot be transcribed or translated into the protein. So, and for this it is very important the obling to occur, because there are 61 codons, and there is only 40 tRNA anticodons. So the number of tRNA is limited. That's why this obling helps in the degeneracy of genetic code. Now, what is the biological significance? It is already described. The codons were not, if the code is not degenerate, I have already mentioned earlier slide, 20 codons would designate amino acids, and 44 would lead to chain termination. So you can see that only 20 codons would designate the amino acids, and out of 3 means instead of 3 termination codons, there would be 44 codons, because there are in total 64 codons are there. This is fixed. So the probability of mutating to chain termination would therefore be much higher with a non-degenerate code. So if if there is no degeneracy of genetic code, then there will be a frequent termination of protein biosynthesis, and this leads to faulty protein synthesis, that is what we call the mutation, and these proteins will not serve the purpose, that is they cannot work properly. That's why mutation will results, and the individual suffers from different deficiency diseases for that particular proteins. We also know that the enzymes are also protein in nature, so that if the enzymes are deficient, then there are so many diseases which can occur. So that's why this degeneracy of genetic code is so much important. So this is all about today's class, and if you have any questions, you can mail me in my email ID, as well as you can send it in the WhatsApp group. So thank you very much.